Welcome to this lecture on the principle of immunology, focusing on autoimmunity. Today, we're going to explore a fascinating but tragic biological phenomenon. We will learn exactly what happens when the body's ultimate defense system, which is designed to keep us safe, mistakenly turns against our own healthy cells. By the end of this session, you will understand the mechanisms behind this friendly fire, how we measure the genetic risks associated with it, and why this knowledge is absolutely crucial for modern medicine. To understand this topic, let us start with a simple analogy. Imagine a highly trained army whose sole purpose is to protect a beautiful city from outside invaders, like bandits and monsters. This army is equipped with the best weapons and is trained to perfectly recognize the uniforms of the enemy. Now, imagine if a glitch happened in their training. Suddenly, the soldiers start believing that the city's own bakers, teachers, and builders are actually disguised enemies. The army begins attacking the very people they swore to protect, causing massive destruction from the inside. In the human body, this tragic scenario of friendly fire is exactly what happens during autoimmunity. Your immune system gets confused and attacks your own healthy organs. Now that we have our analogy, let us look at how things are actually supposed to work. Normally, your immune system undergoes a rigorous education process called self-tolerance. Think of this as a strict background check for every immune cell. During this process, any immune cells that show a tendency to react to the body's own proteins are either destroyed or permanently suppressed before they can do any harm. This central and peripheral tolerance mechanism ensures that your lymphocytes only target genuine threats, like bacteria or viruses, while completely ignoring your own healthy tissues. It is a brilliant system of checks and balances that keeps you safe. So what happens when this brilliant system fails? This brings us to the formal definition of autoimmunity. In scientific terms, autoimmunity is defined as the failure of an organism's immune system to recognize its own constituent parts as self. When there is a breakdown in the tolerance mechanisms we just discussed, dangerous autoreactive lymphocytes manage to escape into the bloodstream. Once they are free, they begin producing autoantibodies, or directly mediating tissue damage through cell-mediated responses. This continuous, self-directed attack results in chronic inflammation and tissue destruction. Depending on which specific organ these confused cells decide to target, this breakdown manifests as various distinct autoimmune diseases. Understanding this breakdown is not just an academic exercise. It is absolutely critical for human health. Autoimmunity is the root cause of more than 80 serious chronic human diseases that affect millions of people worldwide. For instance, in type 1 diabetes, the immune system specifically destroys the insulin-producing cells in the pancreas. In rheumatoid arthritis, it attacks the joints. If we did not study these mechanisms, patients would suffer from progressive organ failure with no hope for a cure. By grasping how and why the immune system makes these mistakes, doctors can develop targeted therapies. Using immunosuppressive drugs or monoclonal antibodies, we can calm the immune system down and give patients a chance at a normal, healthy life. Now that we understand the clinical importance, we must ask, why do some people get these diseases while others do not? A massive part of the answer lies in our DNA. To truly understand how genetics influence autoimmunity, immunologists rely on mathematical models to measure disease heritability and genetic risk. One of the most important calculations we use is called the concordance rate. This metric helps us see the genetic link by studying twins. By observing how often both twins get a disease compared to just one, scientists can determine exactly how much of the autoimmune disease is caused by inherited genetic mutations versus external environmental factors. Let us look at the mathematical representation of this concept. The formula for the concordance rate is C equals N both divided by the sum of N both and N1, all multiplied by 100. Conceptually, this formula is simply finding the percentage of twin pairs that share the disease out of all the affected twin pairs we observe. The variable C represents the concordance rate itself. The term N both is the number of twin pairs where both individuals suffer from the autoimmune disease. The term N1 represents the pairs where only one individual is sick. If the value of N both increases, the concordance rate C goes up, strongly indicating that genetic mutations are a primary driver of the disease. Before we move on, let us see this formula in action with a concrete example. A research team is studying identical twins to understand the genetic basis of type 1 diabetes. They observe that in 60 pairs, both twins have the disease, so N both is 60. In 90 pairs, only one twin has it, making N1 90. We are asked to calculate the concordance rate. First, we substitute these values into our formula. We add 60 and 90 in the denominator to get 150, which is the total number of affected twin pairs studied. Dividing 60 by 150 gives us a decimal of 0.40. Multiplying by 100, we get our final answer of 40%. Physically, this means if one identical twin develops type 1 diabetes, there is a 40% chance the other will too, proving that both genetics and environmental triggers are required. So, we have proven that genetics play a massive role using the concordance rate. This brings us to another crucial mathematical tool used in immunology. Beyond just looking at twins, scientists look for specific genetic markers in our DNA. Many autoimmune diseases are strongly linked to specific genes known as human leukocyte antigen, or HLA genes. But just having a gene does not guarantee you will get sick. To quantify exactly how dangerous a specific gene is, we calculate something called relative risk. This metric compares the presence of the gene in sick patients versus healthy people, allowing us to put a concrete number on the genetic danger. Now that we understand what relative risk is, let us look at how it is calculated. The formula is RR equals P disease divided by P healthy. Conceptually, this is a simple ratio that reveals how much more likely a person is to develop the disease if they carry the gene. The symbol, RR, stands for relative risk. The numerator, P disease, is the probability of finding the genetic marker in people who already have the autoimmune disease. The denominator, P healthy, is the probability of finding that same marker in the healthy control population. If P disease is much larger than P healthy, the RR value skyrockets. An RR of exactly 1 means the gene has no effect, but a value significantly greater than 1 means possessing that gene drastically increases your risk. Let us apply this formula to a real-world scenario. Ankylosing spondylitis is an autoimmune disease heavily linked to the HLA-B27 gene. Clinical studies show the probability of having this gene in sick patients is 0.90. In the healthy population, it is only 0.01. We are asked to calculate the relative risk. We place the high prevalence found in sick patients, 0.90, directly into the numerator. We then place the very low prevalence from the healthy public, 0.01, into the denominator. By dividing 0.90 by 0.01, we arrive at a final value of 90. Physically, this massive number means that an individual carrying the HLA-B27 gene is 90 times more likely to develop this autoimmune disease compared to someone without it, highlighting a severe genetic vulnerability. Now that we have calculated the genetic risks, let us visualize exactly what these genetic vulnerabilities cause at the cellular level. On the screen, you are looking at a side-by-side comparison of normal tolerance versus autoimmunity. On the left, notice the healthy host cell. An autoreactive T cell is trying to attack it, but the regulatory T cell steps in and blocks the attack, maintaining peace. Now look at the right side, representing autoimmunity. The regulatory T cell fails. Without this suppression, the autoreactive T cell releases inflammatory cytokines, shown as red dots, directly onto the healthy cell. Furthermore, you can see a green B cell releasing Y-shaped autoantibodies that lock onto the host cell. This diagram perfectly illustrates how the loss of regulation leads directly to tissue damage. We have covered a lot of ground today, so let us reinforce the most important ideas. First, autoimmunity is a tragic biological error where the immune system loses its self-tolerance, leading autoreactive cells to wage a chronic war against the body's own healthy tissues. Second, we learned that genetics play a massive role in this friendly fire. By using mathematical tools like the concordance rate and relative risk, we can accurately predict who is most vulnerable to these diseases. Finally, remember that this knowledge is not just theoretical. It is the foundation that helps medical researchers develop life-saving immunosuppressive treatments. You now have a solid understanding of the principal mechanisms of autoimmunity. Great job following along with these complex biological and mathematical concepts.